Okay. So, all right. So enzyme substrate specificity. Okay. What ideas have we got about this already from what you wrote? Does anyone want to just share what you understand by this phrase, enzyme substrate specificity? Can you guys see, or do you want me to turn the lights on? Yeah, I don't know. So my grade 12 really like the lights off. I think that's, yeah, because they always like complain they've got migraines or whatever, and they always want the lights off. Whatever. Okay. What do we understand by this idea of enzyme substrate specificity? So we've got an enzyme as a molecule, and then we've got this substrate, which is involved in a chemical reaction that it binds to. This word specificity implies. What do we think?
Specific substrates fit into specific enzymes.
Okay, so there's an enzyme. One enzyme catalyzes every single reaction inside a cell. Okay, so there's thousands, hundreds of thousands of different types of enzymes, all catalyzing one particular reaction with one particular substrate. Okay. So explain. The command term is explain. So that's asking you what? If you're asked in an exam to explain something, what kind of...
Say how it works.
Yeah. The question you're answering is why it works. So explain is always asking you why. So what's a good word to use in your answer? Because. Okay. So enzymes and substrates are specific. Because. Okay.
You don't want to produce undesired products.
There will be waste products, but that's not. There's no intent here. Okay, we're talking about molecules. If I draw an enzyme, okay, it doesn't have a brain. It's not thinking. There's no intent. There's no wanting to. Doesn't really come into it. Okay, what is it about the enzyme that makes it substrate specific?
Its structure.
Okay, that's one thing. What about its structure? So there's two things.
The R group.
What about the R groups?
Well, that's what defines specificity, isn't it?
They're different.
Okay, so let's just remind ourselves. Enzymes are proteins. Proteins are made from what?
Amino acids.
Amino acids. Thank you, Lewis. Great. Sorry, guys. I know it's first thing in the morning, but this is, this is all we've got time for. So, remember, a protein is a chain of amino acids. Okay, that fold. Okay, that because of their interactions between their R groups, that chain of amino acids kind of folds into a particular three-dimensional shape. Okay, so my red line here represents our polypeptide chain. Okay, that's making up the protein. Yeah. Okay, you follow what I've drawn here? Okay, so in sequence along here we've got different amino acids represented. I could represent them by different shapes. Okay. Yes.
Can I take it?
You can come and grab one, sir. Thank you.
Need this one. We could take it or not.
Do you want to write on that?
Yeah.
Well, just grab some lined paper.
Oh, sorry.
Yeah. That looks like grade 12 is lesson work here.
Yeah, that's for.
Okay, so we've got a polypeptide chain that's folded into a particular shape. Okay, it gives the enzyme a three-dimensional shape. Okay, so you can say that enzymes are specific to one or two substrates because, okay, firstly, they have a specific three-dimensional shape. How does that three-dimensional shape affect or mean that an enzyme only binds to one or two substrates?
It's the lock and key kind of.
Good, okay. Thank you very much, Liz. So we have this idea of this lock and key hypothesis. Okay, could you explain that in a few more words? What does that mean? What do we mean by the lock and key hypothesis?
Well, the protein acts as a lock. The enzyme, sorry, acts as a lock.
Yeah, good.
And the different substrates act as keys.
Yeah.
And when that 3D shape, I know we're not going to talk about how it's affected yet, are we?
Not just yet.
Okay,
No.
but that's basically.
So
they bind together.
So what do lock and keys tend to do in the real world, in the physical world?
They match.
They match. Okay, so we have a key, one particular type of key goes in a particular lock. Hopefully, like the key for your front door, right? You know, your key for your front door opens your front door. It's not going to open the person in the flat or the house next door to you. Yeah, ideally. All right. So the enzyme is like a lock. It's like a lock. Okay, this is it's just a metaphor. Okay, that shape means that only one substrate can fit into the active site. Okay, so I could draw my substrate then in green. Notice my active site here is like I've drawn it like a triangle. So my in this diagram, a triangle is going to fit with this enzyme. This is our substrate. But if my substrate was the shape of a square or a circle, hi India, come back please. Okay. If my substrate was the shape of a circle or a square, it's not going to fit. Okay? So that's shape. That's the first thing that determines this enzyme substrate specificity. Okay? But there is a second thing, and that comes onto our idea of R groups that Rhea just mentioned. Let's do the active site. So around the active site we have amino acids. Okay? We have a sequence of amino acids around the active site, and each of these amino acids will have R groups. And what's special about R groups?
They define.
Yep. They define the amino acid,
yeah.
And they're different, and they have different chemicals in them. Yep. So each R group will have different chemicals. So the R groups here determine the chemical properties of the active site. So the other thing is the chemical properties of the amino acids. Can you guys read this? This pen's running out. Amino acids.
What is that? What does that word mean?
Chemical properties of the amino acids in the active site. So do you remember we learned about hydrophobic and hydrophilic interactions? Yeah? We learned that phospholipids have these areas of them which like water and areas of them that don't like water. Okay? We can also have hydrophilic and hydrophobic amino acids. R groups that are hydrophobic, that are hydrophilic, that may have other chemical properties that mean that they might repel certain substrates, but they might be attracted to certain substrates. Does that make sense? So chemical properties of the amino acids in the active site. Certain substrates may be attracted to or repelled. Do you understand this word, repelled? Like pushed away. Any questions about that? Does it make sense? Do you think you understand it, the concept?
So the R group determines whether the key can go into the lock, basically.
Yeah.
Okay.
Think about a magnet. Yeah. Think about what do you when you have like-to-like magnets? What do they do?
They connect.
Like-to-like magnets. So
so they repel.
they push each other away, right? Did you ever play with those toy trains as a
Yeah.
kid? Yeah. So if you've got magnets that are opposite, they will attract each other. If they're so it's the same thing. The R groups in the active site, if they're hydrophilic, they will interact with hydrophilic molecules. But if they're hydrophobic, they might push molecules away. If these R groups have positive charges, yeah, if these R groups were positively charged, they're gonna be attracting any elements that would in another molecule that would be negatively charged. Opposite charges attract. Yeah, but they might, if they're positive charged, they might push away molecules that are positively charged as well. Okay, so yes, the shape is really important, but it's also the chemical properties of the amino acids within that R group itself that determine the enzyme's ability to do its job. Okay. Any questions about that? Go for it, Liam.
Couldn't read like maybe.
Yeah, so certain substrates
Yeah.
may be attracted to or repelled from. What other word could I use instead of repelled? Say pushed away, rejected, yeah, ejected. Okay, cool. So last week we talked about the effects of temperature on enzymes. Okay, this morning I want to talk about pH and substrate concentration, and then we're going to talk about how we design experiments. And you guys are going to be designing an experiment to look at how either temperature, pH, or substrate concentration affects enzyme activity. Okay, so why I've sort of skipped the proteome active because I've got quite a lot I need to fit in the next 45 minutes. Okay, right, let's stop this.